Please welcome to the stage your president, Pia Sundgren.
Hi, and welcome to this morning lecture, the Presidential Lecture. The Presidential Lecture is a lecture that I and my future and coming presidents have the opportunity to choose a speaker that we think could be of value for the society and that could give us a message that we could have and embrace. And this year I have chosen a specific person. I will come back to him in a minute. But the topic that we have chosen and why I've chosen this is related to the fact that we are living in a very changing world at the moment. And we see that there is an increasing mental illness among students and professionals due to anxiety and to stress, maybe because of the increased expectations that everybody is facing. And for us who are seniors, we see a decreasing funding creating a lot of stress. How are we going to keep all our employees and our students in our labs? But also there is a reduction of positions for postdoctoral fellows, which gives a stress to many of those that have finished their PhD and now need to find a postdoctoral position or a permanent position. And also somehow our working environment, both as a student when you study, as well as in the clinical and maybe even in the labs, has been a little bit less friendly and a little bit harder climate. This may be mainly due to the increasing workload we have to deal with. And also there are changes in private life expectations among young people today compared to when I was young. So this year's presidential lecture will be given by Professor Håkan Arheden. He is a professor of clinical physiology at Lund University. He runs one of the most well-known and respected MR cardiac groups in Lund and well-known around the world for this work. But he's also professionally licensed in coaching and mentorship with focus on professionals in leading positions, both in businesses as well as healthcare providers. So why have I chosen Håkan? Well, I have two statements. First of all, I have known Håkan for many, many years. We went to medical school together, and I have seen what he has done over the years, the way he's working with his cardiac group and the environment they have. But also because I have a little what I call hate-love to Håkan. Hate because he has this thing of always force you to reflect on what you say. It's not just to open your mouth and say something. Reflect on what you say and see if you can find a solution to whatever you're complaining about. So that is the hate part. The love part is he does it in such a way that I never have felt intimidated. I have never felt, sad or angry, because I know it's not me he's criticizing, it's what I'm telling, what I'm spitting out my mouth that maybe was not the smartest thing, and that he wants me to reflect on. So please welcome on the stage Professor Håkan Arheden, who will be talking about when stress rules as this year's presidential lecture.
Ladies and gentlemen, President and the organizers of ISMRM, thank you for inviting me to give this year's presidential lecture. This is my disclosure. In 1962, a 44-year-old man was sentenced to lifetime in prison for having led workers in a strike and for having left the country without permission. In 1990, 27 years later, he was released from prison, received the Nobel Peace Prize, and became the president of his country. His name was Nelson Mandela. Just how do people turn the biggest setbacks to the biggest assets? because there are no other constructive alternatives. To mentally resist what happens when it happens is to resist reality. It is stressful and leads nowhere. It is to resist life itself. I will come back to this later. A reasonable amount of stress may help us get out of our comfort zone and perform optimally, whereas too much will impede performance. So what's the relation between challenge and stress? Stress is the gap between how we wish things were and how they actually are. This is why a small gap can be seen as a positive driving force towards our goals, whereas a big gap is no longer seen as a positive driving force, but instead causes stress. I will address stress using 3 categories. The situation, which is on the outside, we on the inside, and how we deal with the situation. The first category is the outside world, which often is described by work, family, world as a whole, and technology. These are given situations that we deal with more or less successfully. The second category is ourselves on the inside. We have thoughts and emotions, but we are not our thoughts and emotions, since we can observe them. We can, for example, turn off our thinking by meditation, and a neutral emotion will follow. That is why meditation results in peace of mind. The third category is how we deal with the outside world. The reality of the outside world is not the same as our perception of it on the inside. Stressful situations occur on the outside, but stress happens on the inside. I'll come back to these categories later. Stress is our body and soul speaking to us with a very important message. It can either be about the outside world, or the way we look at it, or the way we deal with it. Trying to change the world for the better is a good cause. Trying to change it to relieve ourselves of stress may work. Expecting it to change according to our standards, however, is likely not working. Are you sometimes haunted by stress? Then you have a great opportunity to add energy into your career and relations. But that requires change. The key to change is reflective thinking. Reflective thinking means that we think about what happened, why it happened, and how we can improve the next time it happens. And here's the reason to reflect. If we have the same thoughts today as we had yesterday, the results today will be the same as yesterday. If we didn't like the results yesterday, we need to think new thoughts today. If we think new thoughts today, they can lead to new habits. New habits can change our future and eventually save the world. Sometimes one is literally forced by the external situation to make a change in life. This slide shows the outer career development on the x-axis and the inner personal development on the y-axis. The outer career development can be achieved without major reflection. An inner personal development cannot be achieved without reflection. If we're heading for a great outer career development and have no deeper worries in life and suddenly come upon a major stressful challenge, such as being fired, getting a severe life-threatening disease, or being sentenced to prison, as in the case with Mandela, the planned outer career development may no longer be possible. We take a step back, reflect over the situation, evaluate what happened, and reprogram our outer career strategy and maybe life itself. What we dreamt about is no longer possible. Can we have other dreams? We may start an inner personal development where reflection about the life we lead becomes more important than before, when everything seemed to progress smoothly on the outer level. We make other choices, and our inner personal development takes on a new dimension, which usually also improves the outer career development. When I take on new PhD students or young clinical colleagues, they will be trained in leadership whether they want or not. Leadership is about change for the better, and that is why they are trained in reflective thinking. We do this for 1 full hour every week, sitting together, Fridays at 9 a.m. We choose a topic to reflect on. Anyone can suggest it. It could be on how stress affects our lives. It could be on how to achieve a great career or what kind of persons we would like to become. Maybe role models. What's the need for a great outer career development? The short version of the outer career development takes 1 minute. The long version takes 4 years. Let's start with the short version. These 3 traits are necessary for successful outer career development. Deliver on time, be decent, don't badmouth. That's it. Following these 3 traits leads to a great career and less stress. But what happens when challenges on the outside become overwhelming? The ability to deliver on time declines, the ability to stay decent declines, and the tendency to badmouth increases. Anyone who badmouths others can badmouth you. A negative feedback loop starts. So too much stress doesn't help in making a career or having a good life. Let's make a stress test. How many of you always deliver on time? Please, can you raise your hand? 2 persons? Okay. How many of you are always decent? 12? How many of you never badmouth others? 1. Can you feel the tension rise? Stress increasing? Now to the good news. It's really about choice. What makes us human and unique in the universe, as we know it, is our ability to make choices. Active and conscious choices require the ability and willingness to reflect. How does this talk tie into everyday situations? For myself, I love to see my younger colleagues grow into mature professionals and leaders who care about something bigger than themselves. To be successful in their endeavors, and apart from starting with the 3 points on the slide, they need to learn how to handle both stressful situations on the outside and stress on the inside. Those are not the same. But why focus on stress? There are so many other aspects that help growing professionals. Because stress is omnipresent, whether we like it or not. It's present even if we don't focus on it. Stress reduces well-being, creativity, and productivity. Stress wears down the physical and mental health. Some of us are more prone to being stressed and others less so. A young, promising colleague who cannot handle stress will have great, maybe unsurmountable difficulties reaching his or her dream. The term stress in the medical field was coined in 1936 by Professor Hans Selye, who was active at McGill and University of Montreal. His research led to several nominations for the Nobel Prize. Stress reaction results from mental overexertion. In a short perspective, stress reactions can be helpful. Sustained stress, however, causes physical and mental reactions that may be detrimental. Symptoms like tiredness, concentration difficulties, gloominess, forgetfulness may ensue, and diseases as arteriosclerosis, cardiovascular disease, diabetes, depression, and infections may result. And testosterone levels in young men can decline to very low levels after prolonged stress. The term stressor is used to denote something that can be perceived as stressful. This can be something in the outside world or a thought in the inside world. Some stressors are, however, stressing for some but stimulating for others, like parachuting or mountain climbing without security ropes. Some stressors are, however, very frequent and familiar to all of us, and they are usually associated with demanding situations where we have little or no control. Let's take a closer look at the external stressors. Imagine a situation where work was perfect, your boss liked you, your family was wonderful, the world was at peace and at constant temperature, the right politicians were elected, the printer worked, and the phone never ran out of battery. Anyone recognize that as your situation? Then you have ordinary stressors in your life. If they are ordinary stressors, it is not they that stresses us. It is how we look at them, how we judge them. Marcus Aurelius, one of the most powerful Roman emperors and one of the greatest Stoic philosophers, said, "If you are pained by external things, it is not they that disturb you, but your own judgment of them. And it is in your power to wipe out that judgment now." Marcus Aurelius did change a lot of things in the outside world at the time, but the reason was not to relieve stress. To relieve stress, he worked with himself on the inside. The brain cannot separate true from imagined threats. This means that worrying about the future, which hasn't happened yet, one of the most prevalent thought activities of human brains according to research, can be as stressful as real threats. Imagined threats are created by our brain, not necessarily by us. If we don't want to worry about something, but we worry anyway, then it is our brain worrying rather than we. Let's make an experiment. The experiment is as follows. I ask you to close your eyes for 1 minute, a silent minute, and your task is not to think about anything. When the first thought appears in your mind, whatever it is, you open your eyes. Let's start now. Okay, that was 1 minute. How many of you had your eyes closed at the end of the minute? That's less than 1%. So what happened to the rest of you? You agreed not to think, yet you thought. Was it you or your brain coming up with a thought? What you want or plan and what your brain does are not necessarily the same. The brain is constantly scanning the environment for possibilities of threats due to millions of years of adaptation to an environment that is no longer present for most of us. How do we handle that? Let's make another experiment. Close your eyes for 30 seconds and imagine the best scenario you can come up with, an ideal situation for yourself or for the world. It's your choice. Let's start now. Okay, that was 30 seconds. Hold up your right hand and let that represent the ideal situation that you just thought about. And then hold up your left hand and let that represent the actual situation right now. Right hand represents what could be and the left hand represents what is. How's that? Are the hands together or is there a gap? The gap can represent a drive towards the ideal situation, or it could represent stress. What do you choose? You have a choice. What is it? Here we have arrived at the core of this presentation. What's your attitude towards the discrepancy between your dreams and reality? Are you stimulated or stressed? For all sake, keep the dream, but also face reality. I will come back to how we can handle a big gap of stress and make it into smaller, reasonable drives, one at a time. These two experiments demonstrate the power of thinking. According to statistics, a brain that is allowed to think automatically will be thinking primarily negative thoughts, worrying about the future, resulting in negative emotions. So if we take governance of our thoughts, which is very demanding, but possible, we have gained control, at least partly, to one of the keys to control stress. The first key is to understand and choose our thoughts actively. The second key is to train for stressful situations, like pilots, firefighters, or surgeons do. The third key is to understand our choices in a stressful or unpleasant situation. And the fourth is to recover physiologically. Understand and choose your thoughts actively. Automatic thinking is not under our control, and most of it is about worries for the future that hasn't happened yet. Does it help? Likely not. There are several strategies to stop automatic thinking: yoga, mindfulness, meditation, breathing practices, etc. What they have in common is that they help us become aware of reality without thinking, or to divert our thinking to something else. Reflective thinking, however, is under our control, and reflective thinking means controlling our inner space. And since thoughts lead to emotions, controlling our thoughts can lead to controlling our emotions. Controlling our emotions decreases stress. This is difficult and requires training. Elite athletes do it all the time. Train for stressful situations. Control reduces stress. We can either control the outside situations or the inside thoughts. Remember that stress is an adaptation syndrome. It happens on the inside as a result of our thoughts. Challenges on the outside are just situations, and sometimes we just can't control them. But we can train our stress response to become less and less when we expose ourselves to the same challenging situation. When we work with the right level of stress, or maybe we should call it drive instead of stress, then we are in the optimal performance zone. This slide describes 3 psychological states and how these relate to stress. We have ourselves sitting in the comfort zone. In the comfort zone, nothing new happens. When we stretch into the optimal performance zone, located just outside the comfort zone, performance increases and new things happen. If we stretch too far, however, we get into the danger or panic zone, which causes unnecessary stress and performance declines. Another way to view the same phenomenon can be seen in the right panel. At low stress, nothing new happens, and when we stretch into the optimal performance zone, new things happen, whereas at high levels of stress, performance declines again. This scheme is one of several that works pretty well in situations which involve other persons. It is about communication, and communication is one of the core features in biology. Don't act immediately, which most often means don't argue immediately. Listen. Ask clarifying questions. Wait until stress has subsided and reflective thinking resumes. Then get to work. What is happening here is that we are accepting reality slowly. Adrenaline has a biological half-time of about 2 minutes. This means that if we can hold our horses for a few minutes, our brains will go from reptile mode to reflective mode. Repeat this over and over again for as long as it takes. That's what professionals do. Pilots and surgeons do it in simulators. When we think about it, what happens in a cockpit when the alarm goes off? The pilots are trained not to act immediately. then the things in the red box happens. They listen to all information, they go through the checklist and ask clarifying questions. During this process, control increases, stress declines, and then they go to work. Understand your choices in a stressful situation. This might be one of the more important ones, at least when it happens. There are many widely used strategies to handle stressful or unpleasant situations, but only three of them are constructive. The constructive choices are: do something about it, remove yourself from the situation, accept the situation fully. The first two strategies are dealing with the outside, changing the situation as such, or removing ourselves from the situation, whereas the third strategy, acceptance, is about changing ourselves on the inside. Acceptance means to embrace the situation as it is, without trying to change it. It has nothing to do with giving up. Contrary, it is one of the most active and challenging activities a man can engage in. But are there no other options? Yes, but they are not constructive, although used very often, such as to lie, reject, blame, resist, dramatize, badmouth, procrastinate, etc. One way to think about acceptance is the following quote from the Danish philosopher Kierkegaard, one of our most famous existentialists. "This is my life for good or for worse. It is I who perceive the changing circumstances of life. No one else but me can live my life, have my joys and sorrows, even in situations that I personally are not responsible for. It is I and no one else that is there and must make decisions on how to handle the situation." Think about Nelson Mandela, who spent 27 years in prison. What could he do about it? Nothing. Could he remove himself from the situation? No. There's only one constructive option left: to accept the situation fully. Accepting the situation fully meant that he had to work with his joys and sorrows and to change how he saw the situation at hand. By doing so, he also changed the mindsets of his prison guards. Acceptance reduces stress so we can go to work. Recover. Here are the recommendations that our doctor will give us. The most powerful way to recover is to sleep, then short pauses during work, longer rest periods such as weekends, vacations, and in worst case scenarios, sick leave. Always go for some exercise. To the right, we have other examples of exercises that help us recover from stress: yoga, mindfulness, meditation, breathing exercises, and so on. What they have in common is that they divert the brain from automatic thinking. All listed items result in measurable changes in brain activity using functional brain MR imaging. All of this sounds difficult. Maybe we should look at it this way. A 12-year-old kid who dreams about becoming the world champion in a sport understands that this will take years of hard work. How about us? Do we accept that our dreams may require years of hard work? Are we stressed about it? If so, do what professionals do. Break down the big goal into everyday habits and concentrate on performing this day's habit as best possible, starting with this hour and then the next hour and the next. This could be demonstrated with the scale. The weights on this scale are heavily skewed. 1,000 grams in the left pan and only 1 gram in the right pan. And if we were only allowed to put 1 gram a day in the right pan, how many of us would hesitate or give up and spend time on something easier? Here comes the choice again. What is more important in our lives? To invest for the future or hoping for a quick fix? It is up to us. If we choose to invest in a new habit, then one day, when we least expect it, the scale may tip and our efforts pay off. What does that scale represent in our lives, and what will we put in the pans? To summarize, here are the situations on the outside. Stressful for some, but not for others. Here are we with thoughts and emotions on the inside. And here are our options. The constructive options we have are to work on the outside situation by doing something constructive about it or removing ourselves from it. If that is not possible, we have to work on the inside to change our attitude to the situation and see it in a new light. If all of this still is hard to accept, then let me tell you a short story. In 1942, a 37-year-old Austrian neurologist and psychiatrist was deported to Theresienstadt with his wife and parents. He survived 4 concentration camps, including Auschwitz, but lost his wife and family. He became a world-renowned professor of psychiatry, published 39 books, and was awarded 29 honorary doctoral degrees. His name was Viktor Frankl. His most famous quote is from his book, Man's Search for Meaning. Everything can be taken from a man but one thing, the last of the human freedoms, to choose one's attitude in any given set of circumstances, to choose one's own way. That may be valid for stress as well. Thank you.
Thank you so much, Håkan. And with this, I just want to give a little personal present. Keep it, take it with you home. It's safe and valuable. And thank you again for taking your time to come here and give this lecture. Thank you. And with this, I now open up for the plenary session, which will be on MR connecting to other societies.
All right. Now that our lives are not stressful anymore, let's proceed with the scientific program. And I would like to welcome you to the session called Connecting MRI to Other Branches of Science. And I would like to start from saying that in 2018, last year, ISMRM adopted a new strategic plan for the next 5 years. And interestingly, one of its new tasks was formulated to provide opportunities to build relationships and share knowledge with the public with diverse scientific disciplines and with diverse clinical specialties. While we are very good at connecting to each other, engineers, scientists, clinicians, it's now time to connect to other scientific fields, to learn from them, and to tell our story.
Welcome everyone to this plenary. This plenary is devoted to breaking the barriers of a strictly MR community, building bridges and reaching out to those professionals who use other methods to ask similar, if not same questions that we are asking each single day. Ask them how they do it. Ask them what they would like us to do. We think that we have a lot to learn from them, but they have a lot to learn from us too, given the fast development of MR technology. So this plenary is all about listening, being curious, and reaching out. My co-moderator will introduce the first speaker.
All right, so our first speaker is Professor Michael Breakspear. He is a world expert in computational and system neuroscience, and obviously a great connection between our field and neuroscience. He's a senior scientist at Queensland Institute of Medical Research in Australia, and he's now moving to, I think, Newcastle in Australia. And also, conveniently, he is an editor-in-chief of Neuroimage, again, one of the journals that provides the natural connection between us and neuroscience. And his talk is Connecting MRI to Neurophysiology, Collective Brain Dynamics. Welcome.
Okay. Thank you, Dimitri, and thank you very much to the organisers for inviting me to ISMRM and to Montreal, which is a beautiful city. I'm going to run through fairly quickly quite a lot of work and give a case example. So you can see the 4 parts to my talk, some theory, then my sort of day job, dynamic models of large-scale brain activity, a case study of brainwaves, and then to answer the remit that the organisers gave me to speak about more directly the relationship between MR imaging and other forms of imaging and this branch of science. So I have no financial conflicts of interest to declare. Okay, so I'm going to start with a single neuron, which is the basic computational unit of the brain, and show some equations, but the details aren't important. This is a snapshot of the Hodgkin-Huxley model. It's like the seminal mathematical seminal achievement in mathematical physiology. It's a conductance-based model of a single neuron, or at least the soma of a single neuron, and it describes how a neuron spikes due to the interaction between fast depolarizing and slow hyperpolarizing currents. So it's a closed mathematical set of equations which can be simulated and solved, derived from very careful neurophysiological recordings in the giant squid axon. And as an example, this is from a paper by Eugene Isikovich showing the stellar match between this model and the behaviour of actual neurons. And this can extend from spiking to bursting to quiescent to all other types of neuronal behaviour. And it rightly earned the developers the Nobel Prize in Physiology. However, neurons are embedded in circuits which are embedded in functional systems up to the scale of the brain. And this is a bit contentious, but individual spikes are really too weak in all but very exceptional circumstances to influence behaviour. And if their precise timing was crucial to computation, arguably the code would be far too high-dimensional and fragile in the presence of noise. So it seems, to someone like me, invested in the field, increasingly likely that brain function depends on populations of neurons and not individual spikes. Population activity is also matched to the aperture of large-scale brain imaging, fMRI, EEG and MEG. So now I'm going to go through probably most important theoretical notion in this field. So we have a local neural population. This is a beautiful image from a science paper of the hippocampus, not by me. This is a spike raster, which is 1,000 neurons with each spike precisely located along the x-axis. So this, if it was modelled with Hodgkin-Huxley neurons, would be 1,000 times 9-dimensional system. Here's 2 empirical histograms of the states of this population at 2 different times. So the key thing here is to do what's called a mean field approximation, and that is to replace the actual distribution with, in this case, a normal distribution, but it can be another simple distribution. And therefore, we go from a high-dimensional system to a system with just 2 parameters, the mean and the variance, or the value and the precision. Then if we can derive an equation that predicts the forward propagation of this distribution, then we've achieved this central goal in computational neuroscience. And this equation is called the Fokker-Planck equation. And this is just an example of a linear Fokker-Planck equation. And it's a good starting point, but of course there are many more complexities, and that's one of the interesting areas in the field, really. We can also, and it's not unique, and I know there's many physicists in here, and it's the same principle that governs our understanding of the onset of magnetism, supercomputers, LCDs, queuing and swarming behaviour. We can also completely dispense with the variance and just model the mean. This is a neural mass approximation, and we end up with a differential equation from a partial differential equation. So these are the principles of mean field reduction. Even a multi-compartment model does some mean field reduction. We can do further simplification to get a spiking neural circuit model, and this is the field that I'm most interested in, where we lump it all together into a population approach. These are hierarchical mean field reductions, but there's an interesting notion that it's not just a heuristic description, but as in a laser, there's a feedback process or an enslaving principle such that the individual units are somehow slave to the collective motion. This is quite a deep concept in the field, really, and something I find very intriguing. Okay, so here's our evolution equation. It's a mean field equation, and this geometry is the block equation equivalent of my field. So this set of equations in phase space, here spanned by x and y, yields a vector. The set of all vectors is a vector field. The tangent to those yield orbits, and if we look at all possible orbits, due to the work of a brilliant French mathematician Poincaré, we end up with attractors. And the attractor of a system corresponds to the type of time series or behaviour that the system exhibits. And under changes in the parameters, you can go through things called bifurcations. You go from simple to more complex attractors. But there's a one-to-one mapping between the geometry of the attractor and the qualitative nature of the behaviour. I've played around with a very simple neural mass equation which consists of a gross simplification of the Morris-Lacar simplification of the Hodgkin-Huxley, with a very simple inhibitory population thrown in, and some very crude mean field approximations. This is an example of a numerical simulation, and the interaction between the fast and slow timescales of the pyramidal inhibitory neurons can yield bursting, and this is also due to Poincaré. When the timescales are mixing in an irrational ratio, we get something called chaos. So here's one population. Then we go to take a number of these populations and add a few extra terms that introduces the interactions between them. And these interactions occur through white matter structural connections. So on each one, in this particular case, we can put a chaotic attractor, fire them up with as many as you think that the brain should be parcellated into. And in our work, at least in humans, we approximate the connectivity with diffusion-based large-scale connectomes. But we've also done work in primates and rodents using tracer studies. And off we go. So I'm going to show now an example of what occurs, and I'll argue that it's not a particularly isolated example. So we do this. You can download the code from our website and the data, and without much tweaking, you get these large-scale waves. Now, I did applied mathematics, so I happen to love waves. These are called rotating waves. You also get something called breathers, and you also get travelling waves. So these have been described in many different systems, but to our knowledge, using MR-derived connectomics. This was the first description in the human brain, but using biological data. So are these waves the mythical creation of an applied mathematician? In fact, no. Waves have been described in many different species, in many different states of consciousness, and in many different imaging modalities. Why do we get waves? Simply because the connectivity, even of MR-derived connectomes, but also of tracing studies, if we look at distance, On the x-axis versus log of the weight, we get this smooth connectivity kernel, and this is what yields the waves. Here's some ECOG data from epilepsy patients during sleep. This is from the Salk Institute, showing waves of activity at 12 Hertz. And here's our model exhibiting these waves. I'll come back to this in a moment. This is just an arm-waving argument that something is real here. Now, don't worry, the bottom is meant to work as well. So if we, let me try it again. No, I can't go backwards. If we, okay. It did go backwards. There we go. So there's a pattern, a particular quasi-stable or metastable pattern. If we watch it for a moment, it's quite stable. Then something happens. There's some spatio-temporal disorganisation, and then there's a new pattern. So we can plot this using something called an order parameter, and each pattern has a particular fingerprint, and when the entropy of the fingerprint goes down, we can partition the time series into different metastable states. And this is important, but it's also kind of interesting, a way of understanding the dynamics. We can take our model and we can convolve it with a hemodynamic response function. I'll come back to this. And we can see what would it look like in an MR experiment. And we can see and then measure whole brain fluctuations in functional network complexity. And so this is like a forward or generative model of fMRI experiments. Here's some further math. These are the waves. We can do a Hilbert transform and create little vectors, and then we can join them up like streamlines. And in this way, we can go from just observing the pattern to understanding the sources and sinks and the underlying, if you like, dynamic architecture of what we're observing. And when we do this, we see that these metastable states transition when there's a collision or a bifurcation between a saddle and a sink. And if you're interested in the details, you can read the paper. Okay, so that finishes the case study. I'm just going to step back and consider things a little bit more conceptually now. What's the role of brain imaging? So I like this example. Thomas, I can't say his name, wandering around with a measurement device and his postdocs, doing careful observations of the cosmos and coming up with a self-consistent heuristic. Unfortunately, a very complex model which we now know to be wrong. Several hundred years later, some other very intelligent scientists came along, Copernicus, Galileo and Newton, being a little bit facetious here. They used an even more accurate measuring device, but crucially they had an observation model. The theory of optics was absolutely crucial to understanding and breaking the prevailing theory of the cosmos. And also with, of course, Newton developing the calculus. And so this is kind of correct. Of course, we know it's not a complete description, you need relativity. So here we are in the neuroimaging community, doing observations and experiments and observing an ICA mode using Steve Smith's tool. But we need to understand that there's a measurement process here, a hemodynamic response function. And so this is the mud map of computational neuroscience. So in order to do a theory of the brain, we have to understand we're inverting this process here. I'm nearly finished. Here's a particular example of our neural mass model. We have a hemodynamic forward model. We can then predict and compare static and dynamic functional connectivity, or we can use a biomagnetic forward model and we can predict and measure the dwell time distributions of these waves. And working with Mark Ulrich in Oxford, we did this with MEG data, and the predictions are very accurate. We can also use ECOG data and a dipole model and understand the propagation speed and wavelength and measure those. So in summary, what can imaging do for large-scale models? It can provide or parameterise the models. Here, connectivity, and a very exciting development is using myelin density as a proxy for timescale parameters. And imaging can also yield time series data against which we can test our predictions. What can models do for imaging? Well, they can provide causes or mechanisms of the observed empirical data. Models can also integrate multimodal data. I showed an example of fMRI and MEG, but we have also worked to integrate physiological data, and I'm also very interested in collective human behaviour. There are now open source simulation engines. This one's a Python one, written by Vic Yersa, Petra Ritter, and Randy McIntosh, and Nancy Lebeau, who's here, and the team, and this is a MATLAB-based one. You can download all the code and the data for these simulations from our website, and I'd like to thank my team here that I left behind in Brisbane and our funding sources. So thank you.
Thank you, Professor Breakspear. That was an excellent talk. I am going to invite our next speaker, Professor Lotario Toro. He was born in Chile, trained as a mathematician, and then went on to do his PhD in computational and fluid mechanics in the United Kingdom. Today, his methods are used worldwide in a broad range of branches in science and engineering. He has a long experience in applications in aerospace sciences, industrial mathematics, and environmental disciplines. He is recipient of several honors and distinctions, which include 2 doctorate honoris causa, the William Penny Fellowship at the Physics Department of the University of Cambridge, and the honorary title OBE, Order of the British Empire, conferred by Queen Elizabeth II for his meritorious services. Professor Toro is currently Emeritus Professor of Mathematics in the University of Trento, Italy.
Thank you very much for the invitation to speak to this very distinguished audience. So my topic here is computational methods to compute the dynamics of fluids in the full human body. That is the idea. Now, how do you go forward here? How do you go forward here? Which button is that one? That one. Well, nothing to declare here. Now, I shall begin by describing our approach. We began with fluid physiology. This contains, embodies the biophysical mechanisms underlying many diseases, in particular neurological diseases. We take a holistic view of the problem. That is to say, we consider the full human body with regards to fluid dynamics. We consider all relevant fluid compartments, and when relevant means you make some choices here. And we also consider the anatomical and functional connections of these districts. This naturally leads to a multidisciplinary approach, where we have the contribution of fluid mechanics. Imaging comes in, of course, in a very natural way here. We have mathematics and computing, and obviously biology, experiments, measurements, and medicine. Now, for this talk, the fluids of interest, the fluid systems of interest are the following. We began with the arterial system, of course, very much studied, conventional. In fact, even from the point of view of computational fluid mechanics, a lot of work has been done in the last few decades, in fact. The venous system is much less studied. It has received much less attention. unfortunately. And then we have, of course, the connection between these two systems, the microvasculature and the pulmonary circulation. Also less studied is the lymphatic system. And here we see at the level of the microvasculature, all the relevant fluids of interest to us, the venous part, the arterial part, the interstitial fluid, and lymph. If we want to study cerebral hemodynamics, we must include also the cerebrospinal fluid, of course. Some remarks on the venous system. Now, we all know it returns oxygen-depleted blood to the heart, but it also plays a major role in clearance waste from the body. It drains ISF, CSF, and lymph, holds about 70% of the blood in the body. There are high individual variabilities from person to person that makes the study of the venous system complicated. And it plays a major role in CNS dynamics generally. It is highly deformable. So veins collapse even under physiological conditions. You can see here on the right-hand side the behavior of the cross-sectional area of a vein as a function of pressure. And you can see that at high pressure here, you see essentially a circular cross-sectional area like in arteries. But as the pressure decreases, they begin to deform, and you have even the situation of collapsing. As a matter of fact, in about 2/3 of the normal population, in the sitting position as you are, internal jugular veins are actually collapsed. But of course, you don't need to panic about this because you have the vertebral brains that would do the job for you to take the blood from the brain back to the heart. Now, when it comes to the venous system, we have a number of situations that need to be addressed, in particular the question of anomalies in the venous system. Here I am showing you a healthy control, and we see the main veins here, for example, the superior sagittal sinus, the straight sinus, transverse sinus, sigmoid sinus, are all leading to the jugular veins that will take blood from the brain back to the heart. And anomalous cases on the left-hand side there, you can see that that patient, compared to the healthy control on the right-hand side, has a very anomalous venous system, fundamentally in the intracranial system. But also you have extracranial venous anomalies, and this is the study done by Haki and collaborators. And you see here a range of situations in which internal jugular veins display anomalies. Now, talking about anomalies of the venous system, you have then association of this to a number of diseases. In the last 10 to 15 years, a number of groups around the world have put forward some theories about the possibility that these diseases of the venous system could be associated to neurological diseases. For example, idiopathic intracranial hypertension, multiple sclerosis. In ophthalmology, you have similar situations. Meniere's disease, in the last few years, there's been a lot of work in this area, even in idiopathic Parkinson's disease. Now, these are the situations, it's a situation regarding major fluid systems in the human body. Now, let's do a little bit of mathematical modeling, which is the main part of this presentation. So in 2014, we published our paper on a global model for the human circulation, including also fluids in the cranial cavity and the spinal cord. So 5 years later, we produced an updated version, is the one I'm talking about today. And what you can see here is the representation of the arterial system, the venous system on the left. We have in between the microvasculature, we have the heart, the pulmonary circulation, and the model for respiration. When it comes to the head, in addition to the blood districts, of course, that we have to include, we have the model for the cerebrospinal fluid. including all ventricles, the subaryanoid space in the cerebral part of the body, and also in the spinal cord. Now, just to give you a flavor of the mathematics behind this model here, we are showing the differential equation here for a single vessel. And you have there, on the left-hand side of those equations, you see the unknowns, the ones which are differentiated with respect to time. So you have the cross-sectional area of each vessel, because we are talking about compliant vessels here. And we have the flow, which is the product of the cross-sectional area times the velocity. Now, this equation has to be solved, obviously, numerically. And then they are applied to all vessels in the body which are part of the model, veins, arteries, and so on. The unknowns, I just said it, are the cross-sectional area, which depend on position along the vessel and time, the flow, and the pressure. So given the solution of this system of equations in the full body, we will have all these quantities as a function of position and time. Now, this is a demonstration of the performance of the method with regard to the interaction of fluid compartments. You see in the brain, in the cranial cavity, you see the interaction of the arterial blood, the red rectangle, the venous blood, and the cerebrospinal fluid within each cardiac cycle. So obviously, as a high volume of arterial blood comes into the brain, because the total volume has to be constant, then it means that other materials have to be ejected from the brain. And the other materials are the venous blood that has to come out, and the cerebrospinal fluid that goes down into the spinal cord. So this dynamics is very important. If you disturb it, you can understand by looking at this picture, if you disturb the exit or the outflux of the venous system, for example, you are going to alter the dynamics of these fluids, which must work in concert. Now, the fact that in the previous picture the situation seems to be working, it doesn't mean that it's correct. So you have to do a very painful exercise of validation of your mathematical model here, and we do this through a comparison with magnetic resonance imaging measurements here. So what you see here is a comparison of computations for the right transverse sinus here. And you can see this is within the cardiac cycle. So you can see that for flow on the right-hand side of the picture, you have 3 curves. 2 curves correspond to 2 versions of our mathematical model, and they agree pretty well, which means that at least we are agreeing with ourselves. And then the third one is the measurement from magnetic resonance imaging. And the agreement between theory and measurements is pretty reasonable in my view. You can also obviously do a lot of work on the validation here. Here we have again another comparison for other venous vessels of the head and neck. Here, because it is a global model, we can also do the same exercise for the arterial flow in various compartments here. And again, the comparison of our simulations with existing data is quite reasonable. Now that we have a tool, we can actually use this tool to study some problems. And in particular, we would like to do some work on predicting the impact of venous outflow strictures on the general dynamics of fluids within the brain here. So we consider here 2 cases, A and B. The case A has a stricture in one of the internal jugular veins and the aspiragus vein, and the right-hand side is case B with 2 occluded jugular veins. And we see here on the left picture there a comparison for pressures between the anomalous cases and the normal case as a function of time within the cardiac cycle. And we see that both anomalous cases have higher pressure. In other words, we are predicting venous hypertension produced by outflow strictures in the neck. Now, these are predicted values for the cerebrospinal fluid pressure in various compartments in the brain and the spinal cord. For example, you take, let's take the fourth ventricle, for example. In the fourth ventricle, which is 4V, you see two curves here as a function of time within the cardiac cycle. So the black line shows the normal case, the red line shows the anomalous case in which there are strictures to the outflow of blood from the brain through the internal jugular veins. A similar situation can be seen for the remaining compartments of the cerebrospinal fluid, including the spinal cord. So our findings can be summarized as follows in the kind of physiological theorem, if you like. And we say that if there are cerebral outflow strictures intra or retrocranial, then there will be increased cerebral venous pressure, disturbed CSF reabsorption, increased CSF volume and pressure, increased intracranial pressure, ICP, and in general, disturbed cerebral venous return to heart. Two messages. I would encourage you to adopt a holistic view of bodily fluid systems. So if you are interested in studying, for example, the cerebrospinal fluid, it is very important that you connect these to other systems, particularly the venous system. Now, the venous system is crying for your attention. New developments, I could not finish this lecture without mentioning an important development in fluid physiology regarding the central nervous system. Working in Southampton with the iPad system, the lymphatic system, we've seen a lot of presentations in this conference here. The meningeal lymphatic system, of course, a little revolution in this area here. Obviously, there are a lot of issues with these theories and a lot of questions to be answered. And so mathematical modeling is very uncertain, very difficult. But we are making an attempt here in collaboration with the Virginia group of John Guinness, and we produced a sort of working model for an animal, and we have some preliminary results. Now, the connection between what I do and what you do here, well, essentially, the way I see it, it is a one-way relation here that mathematical models need magnetic resonance imaging to begin with for determining the computational geometry. Then, number two, we also need measurements in order to validate our mathematical models. So essentially, we are using you to do our work. Highly desirable would be to compute pressures, non-invasively, of course, by a combination, perhaps, of magnetic resonance imaging and mathematics. There is some work in that direction, which is interesting. A final possible two-way interaction would be to put together MRI and simulations of fluid dynamics in order to assess our own errors, both in MRI measurements and also in simulations. I conclude. I have pointed out the major extracellular fluid compartments in the body, their interaction, our holistic approach, a mathematical modeling, perhaps leading to a virtual human in terms of fluid dynamics, applications to assess the role of the venous system, and perhaps an interaction between modeling and imaging. Challenges, of course, ahead, but also great opportunities. Thank you.
Thank you very much, Professor Tora. And our final speaker is Professor Mikhail Shapiro. He's a professor of chemical engineering at Caltech. And Mikhail is an expert in molecular engineering and genomics, and he is a great source of connecting our field with this basic advanced biological methods and tools. So his title is Connecting MRI to Molecular Engineering, Genetically Encoded MR Reporters. Welcome.
Great. Well, thank you very much to Dimitri and Nivedita for inviting me. It's really a big pleasure and honor to be here and to help us step a little bit out of our comfort zone into these new topics. So the dream that drives a lot of the work we do in our lab, and I think more broadly the field of synthetic biology, is the idea that in addition to drugs that we take that go everywhere and cause side effects, or surgery where we have to cut somebody open with a knife, we're going to have smarter medicines in the future that are going to be little microscale or nanoscale devices that are going to be roaming our bodies and sensing pathologies and treating diseases very locally. That are going to be smart. And of course, this idea and this image here is shamelessly ripped off from sci-fi, from the movie Fantastic Voyage in 1966, starring Raquel Welch, that I'm sure many of you have seen. And so, the interesting thing is that this vision is actually coming true, but with one modification, which is that instead of mechanical and electronic devices that are shrunken down to the size of a cell so they can be inside of our bodies, we're using cells themselves. And the reason for that is if we think about what we want these devices to do, to roam through our bodies, home to sites of disease, take in molecular signals from their environment, make decisions based on those signals, and then execute therapeutic programs like releasing proteins or killing other cells, cells already do 90% of that. And so the idea is that we can leverage what cells already know how to do and then add in this additional 10% that allows them to do that at our direction. And what's making this possible is within this field of synthetic biology that's developed over the last 20 years or so, is that we now have really good ways to engineer and program cells. So this here is one of the first examples of that. And one level at which we can engineer cells is through their genetic program. So we can have genetic circuits that obey differential equations, more or less, that allow us to program in logic and computation into cells. The other thing that makes this possible is that we can give cells new functionalities by engineering and endowing them with proteins that can do new things. For example, receptors that we can put into immune cells that instead of recognizing antigens that they themselves learn to recognize, can have them recognize antigens of our choice. And for example, use that to direct immune cells, these green guys, to go and recognize and shock and destroy tumor cells. And based on these capabilities, we've had some really beautiful recent advances in cell-based diagnostics and therapeutics. And I'm sure you've all heard of things like CAR T immunotherapy, where you can have a cancer patient take out their immune cells, genetically modify them to express receptors that more selectively recognize the cancer that that patient has, send those cells back into the body, have them go and kill the tumor. This is FDA approved in the last year and a half. Another example is engineered probiotics. These are bacteria that we eat in our yogurts, but now have been genetically modified to do things like detect inflammation in our gut and release anti-inflammatory compounds. Now, one of the challenges within this field is that we take these very sophisticated agents and we inject them into our bodies, but currently we don't have good ways to see where are they. Are they alive? Are they executing the functions that we programmed into them? And we don't have very good ways to tell them what to do based on their location. Of course, and that's the problem that we need to solve as a field. Now, we do have great ways to do that under a microscope. Right? So the cells I showed you earlier were expressing green fluorescent protein, which is something that was cloned from jellyfish. And if you put this protein, or the gene encoding it, into cells of your choice, you can see all kinds of beautiful stuff. You can watch cells dividing, expressing genes. You can see subcellular structures. The rainbow images we saw in the earlier talk came from these kind of fluorescent proteins. And so, of course, this is limited because light doesn't penetrate very well into tissues. So the question is, can we do something like this with MRI? And this is not a new idea. In fact, over the last 20 years or so, we and others have thought about, is there a GFP for MRI? And the way we started out doing this was saying, okay, well, let's look at our synthetic contrast agents, paramagnetic chelates or superparamagnetic iron oxides, and look for proteins that look similar. Okay, so naturally, we turn to things like ferritin or transferrin, which help cells take up and store iron, cytochromes, which can chelate iron. And that worked to some extent, but I think it's fair to say that these technologies have not yet become widely used by even the basic biology community. And that's because their performance is not that great, if we're being honest with ourselves. And the main limitations for that are sensitivity. So you need tens of micromolar typically of these things to see them, which is not trivial for a cell to make. And then if you're using metals, there are issues with metal availability. And so I want to show you a couple examples of how we and others are thinking about taking alternative approaches to this that perhaps can help us do a little bit better. So one idea is to turn to diffusion-weighted imaging. So the agents I showed you earlier were based on T1 and T2. There are also ones that are based on CEST. But diffusion, is one of the main forms of MRI. I think there's a huge contingent of people at this conference who are working on diffusion. And so is there a reporter gene we can make for diffusion-weighted imaging? And this actually turned out to be very straightforward once you think about the fact that one of the main determinants of diffusion contrast is the ability for water molecules to get across the cell membrane. And so all we need to do is to have a protein we can put into the cell membrane that'll alter its permeability, obviously in a healthy and well-tolerated way. And nature has a gift for us in the form of aquaporin. This is a transmembrane channel that is exclusive to water. It's passive. It doesn't pump water in or out. It just lets water molecules go out and come in, increases their diffusivity across the membrane. Each aquaporin can conduct up to 1 billion water molecules per second. So it's really a remarkable protein. And there had been some previous studies connecting endogenous expression of aquaporin to diffusion-weighted imaging. And so the idea we had was very simple, is that if we overexpress aquaporin in cells, it would make the membrane more permeable and cause contrast on diffusion-weighted imaging. And if that's true, then this is a reporter gene that cells can make that does not require any kind of metals. It's a small autologous gene, so it should be well tolerated by the immune system, and the mechanism is orthogonal to the other reporters, so it could be used in combination. And luckily, this turned out to work really well. So here we're looking at some data where we have cells, these are pelleted cells, 3 different cell types, that are genetically modified to either overexpress aquaporin or to express green fluorescent protein as a negative control. Under diffusion-weighted imaging, the cells expressing aquaporin look darker, as expected. And so now that we have that, we can connect it to a simple genetic circuit. So here we have a drug that's driving this promoter that's going to make, as a function of how much drug we put in, aquaporin on these cells. And what we can see is as we increase the quantity of drug, we see our cells that are programmed to express aquaporin in the circuit getting darker and darker and darker, and we have this expected transfer function between our chemical input and our signal output that we can see by MRI. This is an experiment that's been done thousands of times with fluorescent proteins, but now we can see it using our non-invasive technique. Of course, we had to think a little bit about MRI to make this work. If you just use the type of pulse sequences that most people use with short diffusion times, actually turns out not to work very well. Dimitri could have told me that in his sleep. And so we had to go to longer big delta sequences using a stimulated echo, and that's what produced the beautiful images you saw on the previous slide. This also works quite well in vivo. So just as a model system, we have a couple tumors that we're implanting. One will be triggerable with a drug to express aquaporin. One will be triggerable to express GFP. Before we give the drug, both of them look bright on diffusion-weighted imaging because the tumor is highly cellularized. But when we give the drug, the tumor that's programmed to express aquaporin in response to that drug turns dark. Okay, so we can see gene expression here in the living, breathing mouse brain. And I want to acknowledge that we weren't the only ones to think of this idea. Simultaneously, there's a publication from Kevin Brindle's group on using a urea transporter, which in addition to urea can transport water, and they saw a similar kind of contrast using a slightly different pulse sequence. Okay, so that's using a fairly simple protein. We can produce nice MRI contrast. I want to talk about something a little bit more exotic. And so I already mentioned that we and others had looked at T2 contrast agents based on metalloproteins. And the key limitations there have been that metals may not always be bioavailable. And then with T2 contrast, there's also this problem of background contrast where you can't tell if the contrast is coming from your reporter or something else that's present, like blood bleeding or other kind of phenomena. And so we asked, can we develop a biomolecular T2 reporter that, again, does not require metals and can be distinguished from background T2 contrast? How are we going to do that? So we had this kind of wacky idea. How about a protein filled with air? Okay. The logic behind this is that the magnetic susceptibility of air is different from water. If we take a nanoscale object, that's filled with air, put it in a strong magnetic field, then we're going to generate little field gradients around this nanoscale object that should produce T2 contrast. And if we have a larger volume that's filled with some amount of these contrast agents, it's going to change the macroscale susceptibility of that region and produce quantitative susceptibility contrast. Okay? So all we need is a protein filled with air. Now you might say, what the heck? Where are we going to find a protein like that? But one of the key lessons of synthetic biology is that nature has all kinds of gifts in store for us, because it's had to solve all kinds of problems throughout evolution. And in this case, we found what we needed from these organisms. These are the South Bay salt ponds near San Francisco, where the color in these bodies of water comes from photosynthetic microbes that had to solve the problem of regulating their buoyancy, so they can always be in the right level in the water to receive enough sunlight for photosynthesis. And the way they evolved to do that is by forming these really beautiful intracellular protein nanostructures. They're called gas vesicles, but it's actually a misnomer because there's no lipid. It's completely a protein shell. And what's special about them is that this is one of those structures that we've purified from the cell. You can see it's a couple hundred nanometers in size, and it's hollow on the inside. You have a 2 nanometer thick protein shell that encloses an interior compartment that's just full of air. And the way it works is whatever gases are dissolved in the surrounding media can partition in and out according to their Oswald coefficient, just like at an air-water interface. But water is prevented from coming in and condensing into a liquid by a very hydrophobic interior protein surface. And for these cells, when they want to float higher, they turn on the genetic program that encodes the proteins that assemble into these nanostructures and make more of them, so the cell density decreases. And when the cell wants to sink, they just break these things down with proteases and sink back down. And so here we have a protein cell can make filled with air. Okay? And we hypothesized this would be able to produce contrast in T2-weighted and QSM imaging. And luckily for us, that hypothesis turned out to be correct. So here we're looking at a concentration series of these purified proteins. We can see contrast by T2 and T2 star imaging, and we can also see QSM contrast. And it turned out to be quite sensitive. So the detection limit as of this paper was about 250 picomolar of these protein nanostructures, which corresponds to 25 micrograms per milliliter of protein. Now, for reference, a typical cell makes 150 milligrams per milliliter of protein. So we're only asking it to make a small amount relative to its capacity. Now, another problem with T2 MRI that I mentioned is that we have this background contrast. And here we illustrate that by having a sample where we have a couple of these wells that contain some of our gas vesicles, but we also have just different concentrations of agarose, which themselves can produce T2 contrast. And we purposely put a little rod full of nickel in the background as a metal artifact. Okay, so here it's very hard to distinguish where the gas vesicles are from everything else that's there. But here we can take advantage of another feature of these nanostructures, which is that they are acoustically erasable. And what I mean by that is that, it's a thin shell, and we know from other studies that we've done in the lab that if you apply ultrasound where the amplitude of the sound wave is above a critical collapse pressure, then you can cause these gas vesicles to break. This is what it looks like when it's broken. And the gas inside of it dissolves within about a millisecond and is gone. Okay, so the susceptibility change should be gone as well. And the way we can take advantage of this is we can take this confusing image, take an MRI image, apply ultrasound. Okay, you can see that there was a slight change in these two wells here that contain the gas vesicles because they've collapsed. And we do a subtraction, and the difference image shows us this very nice contrast that's specifically due to these nanostructures. So it's a way to combine MRI with ultrasound. And here, the ultrasound requirements are actually very simple. You're not doing any imaging. You're just having a sound wave that's coming through. The frequency more or less doesn't matter, and it doesn't need to be focused. This works not just in phantoms, but also in living animals. Here we're doing this in mice. We've injected some gas vesicles into the spot. Looks dark. We apply ultrasound. Bam. Signal disappears. Then we can do a subtraction, overlay it on an anatomical image, and we get our nice specific contrast there. So this is with purified proteins, and so now we want to put this into cells. And so the task that actually turned out to be quite challenging is to take this dozen genes that allow the cyanobacteria to make gas vesicles and port them over into other cell types that we're interested in. And we started doing this with commensal bacteria that we want to study in the GI tract and other places. And after some genetic engineering, we now have this synthetic gene cluster that we can put into E. coli. And here you're looking at a TM image of an E. coli that's just full of these little gas vesicles inside of it. And now we can see gene expression in these living cells with MRI. Okay, the last thing I want to talk about with MRI is, can we push the limits, right? We want to be as sensitive as possible. And so one of the things that I know a number of people in this room work on is hyperpolarized MRI, which is a great rejection of thermal equilibrium, and putting spins into a transient non-equilibrium state of enhanced polarization. And one of the attractive ways of doing that is with noble gases. Xenon, for example, can be polarized with spin exchange with a rubidium vapor. And then it can be inhaled and distributed to tissues, and they're pioneers of this technique here in the audience. Now, one of the things that allowed this, the idea is that you could get orders of magnitude higher molecular sensitivity from this. Now, the thing that connected this to molecular imaging initially was some work from Leif Schroeder and Alex Pines, showing that you can take these beautiful glove-like compounds that can bind xenon and do chemical exchange saturation transfer detection, where you irradiate at the chemical shift of the xenon inside this cage, have it exchange with the bulk pool, and thereby reduce the bulk magnetization. Okay, so you can get very sensitive detection of these chemicals, not at micromolar concentrations, but at sub-nanomolar concentrations. And one of the criticisms, of course, is, yeah, but you have these huge synthetic compounds, how do you get them into cells, into the body? So could we make a protein that can interact with a gas like xenon? And you will not be surprised that our solution to that was gas vesicles, right? Because we have these proteins that a cell makes that allow whatever gas is dissolved in the surrounding media to partition in and out. And it turns out that you can do hypercest imaging with the gas vesicles and thereby bring down their detection limit now to 1 microgram per mil. So an order of magnitude lower than what we had previously. And you can have several different types of gas vesicles that have distinct chemical shifts that lets you do a form of multiplexed imaging. Okay, the last thing I wanted to mention in the 1 minute I have is another role for MRI in synthetic biology is to help us tell cells what to do. Okay? Now, MRI itself, there are people working on magnetic actuation, but MRI itself, in this context, is doing imaging. But we can couple it to modalities, that allow us to perturb cellular function. So one of the things that we're doing in our lab is that we're using focused ultrasound to tell cells what to do by engineering genetic programs that get turned on upon ultrasound irradiation. Here, this is implemented in bacteria with this genetic circuit that responds to small temperature elevations that focused ultrasound can impart to a millimeter scale location in the tissue. And the role for MRI here is to dial in and monitor this temperature elevation. Because a lot of people are doing high-intensity focused ultrasound for ablation, where maybe you don't need to monitor quite as carefully. But if you're just trying to tell cells what to do and have temperature that's going to be within the range of 37 to 42 degrees, you really need to see what you're doing. And so here we're combining this focused ultrasound with real-time MRI thermometry that allows us to essentially paint patterns of gene expression using MRI-guided focused ultrasound. Okay. Thank you very much for your attention.
All right, so this concludes our session. We heard 3 remarkable presentations connecting us to very different fields. Let's give all 3 speakers a round of applause.